This is the first lecture for Friday, April 17th. And for this lecture, we're continuing on with our survey of marine ecosystems. As you recall, spent all this time looking at different coastal ecosystems. And then we moved on to the open ocean. So we've covered huge parts of the ocean. And now we've shifted our attention to these specialized marine ecosystems, specialized in the sense that life there is not as easy as some of these other places, even though some of these other places are relatively easy to live. Some of what we focused on were adaptations that organisms have to live there. If you remember that organisms that live in estuaries tolerate rapid and drastic changes in salinity, especially intertidal organisms tolerate being in and out of the water. And in the deep sea and in the polar ecosystems that we look at soon, well, there's even more stressful conditions. It's even harder, more extreme environments in terms of harsh environmental conditions. And when we're looking at the deep sea, in fact, we saw that there's no light. There's adaptations that organisms have for that. It's high pressure and very cold temperatures. Adaptations that organisms have in respect to surviving under those conditions. And then the last thing that we're looking at was this idea that there's low biomass in deep sea habitats. Now, if you added up all the deep sea habitats, there'd be a considerable amount of biomass, but there's no primary production there other than the tiny little bit that occurs as a result of chemosynthesis. Everything else comes from the surface. And we saw that there's a number of adaptations that organisms have in response to this overwhelming condition of low biomass or relatively little to eat, little amount of food. And we saw that one of the overwhelming adaptations in deep sea organisms was neutral buoyancy. There's a very, very strong selection pressure for organisms being neutrally buoyant. If you're not laying on the bottom and you're in the water column, then there's strong selection pressure for being neutrally buoyant to be able to maintain your position in the water column. You can't constantly swim searching for food, staying afloat, trying to find something to eat. There's just not enough energy there for you to consume to support that lifestyle. So what I want to talk about today is this specific group of deep sea animals, deep sea sharks, and the adaptations that they have towards neutral buoyancy. There's strong selection pressure for deep water sharks to obtain neutral buoyancy, just like there is for other fish, crustaceans, mollusks, things that are in the water column, salps. And with sharks in particular, we're going to see that their solution as a whole, if you look at a typical deep sea shark, they're neutrally buoyant. And they achieve this neutral buoyancy through three different mechanisms. One, that they have a large liver. Two, this liver is really, really oily. It has a really high oil content. And the oil that's in these livers of deep sea sharks has a high concentration of these special low-density lipids. Diacylglycerol ether, which is DAGE, and squalene. Now, there's some other organisms out there, some zooplankton and some plants that produce diacylglycerol ether and squalene, but it's not very common. These are unusual lipids, and you find them in large quantities in deep sea sharks, and not very many other things. So this is what the typical deep sea shark does to attain neutral buoyancy. Large livers. If you look at the liver size of deep water sharks and compare that to the size of the liver of other sharks. Now, sharks in general have relatively big livers compared to other organisms. In a typical shallow water shark, a liver might be about 5% of the body weight. If we did a search for the size of liver in humans, it's a big organ, but it's not even 5%. If you look at deep water sharks, so in this figure, there's deep water sharks and these yellow dots. The species, different species, are listed on the x-axis of this, and the y-axis of this figure is percentage of body weight represented by their liver. So within that circle are deep water sharks, and you notice that the size of these livers is 20%, 25%. These are really big livers. A quarter of their body, or a fifth of their body, is made up by these livers, big livers. And if you compare that to other organisms, that's not the case. So they have a big liver. And organisms, well, deep water sharks that have big livers are neutrally buoyant. Now, this figure shows the density. So if you have a density of 0, that means the density of your body is equal to the density of the water that there is. So there's no difference between the density of your body and the density of water. You're neutrally buoyant. If you're heavier than that, then you have a density of 2, 4, 5, which is typical of other species sharks. So you notice all these yellow dots. Those are all deep sea sharks. They're all close to neutral buoyancy. And the x-axis is how big their liver is. So a big liver in these deep water sharks is certainly associated with neutral buoyancy, or a density essentially of 0. Second feature of deep sea sharks in general is that they have a large amount of oil in their liver. A lot of oil, oily. Now here's a picture of a shark, a deep water shark, that was cut open, and its liver comes pouring out. It's hard to imagine being able to stuff that liver back into its body. It's so big, 20-something% of its body weight. And another thing you can't see in this photograph, but if you were watching this, you'd see oil oozing out of this liver. It's so full of oil. In fact, if you look at the amount of oil that's in the liver of these deep water sharks, it's typical to have a liver that's 80% oil. 80% of the weight of this liver is made up of oil. Now that is really oily. If you look at a typical shark, a shallow water shark, their livers are fairly oily. And in fact, they've been harvested for oil, these shallow water sharks, but maybe it's 20% or so of the weight of their liver is made up of oil. And these deep water sharks, you can see in this oval, the red oval, here's these different species of deep water sharks designated by some abbreviation. The amount of liver oil is close to 80%, over 80% for some of these species. Now that is a lot of oil. That is very oily liver. So for deep water sharks in general, they do have a very big degree, a very large degree of oil in their liver. So a large liver and a large amount of oil in the liver. This third concept is that the oil in this very oily liver, in this very big oily liver, contains these classes of lipids, these types of lipids that are unusual for one, but they're also very low density. So let's look at these livers and these oils and these low density lipid classes. This is diacylglycerol ether and squalene. So here's what these two low density lipids look like. The top one, squalene, it's a hydrocarbon. It's just got hydrogens and carbons. Carbon, 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 50 carbons or so, 55 carbons with a bunch of hydrogens. That's that red chain up there at the top. The density of squalene is about 0.86. Now, that might not mean anything to you, but if you think something that everybody knows, certainly, is the density of water pure water is 1. Seawater is more dense than pure water. So this density of 0.86, that is really low. The next one in the blue is diacylglycerol ether, which has a density of about 0.89. This diacylglycerol ether is very similar to triglyceride, which is the most common lipid in animals. It's got this glycerol backbone on the left that's at CH and CH and CH. If you look at triglyceride down on the bottom in the green, that's a glycerol, 3 carbons with hydrogens, and 3 fatty acids coming off, ending in the R group. It's a certain number of carbons that come off there. So we looked at this little with phospholipids when we were talking about cell membrane fluidity. So this small change in the fact that you've got an ether in the very top of that diacylglycerol ether, that CHOR is an ether, as opposed to a 3 fatty acid change you find down on triglyceride. That small substitution results in a different kind of lipid that's much less dense than the typical triglyceride that's so common in animals. 0.92, still pretty low density compared to water, but it's higher density. It's not as, doesn't provide as much buoyancy as the simple substitution with an ether for one of the fatty acids that results in a density of 0.89. So if you load up those lipids in your muscles, in your liver oil, in other parts of your body, but particularly in your liver oil, that results in a pretty significant increase in the amount of buoyancy that your liver provides. It's not just that you have a big liver. It's not just that you have a big liver that's got a huge amount of oil in it. It's that you have these low-density lipids, diacylglycerol ether and squalene, that are in large quantities. So if you look at some other lipids that occur in animals in general, in us, in humans, the most common lipid that you find is triglyceride. Has a density of 0.92, 0.93, around there. It depends on the number of carbons in the fatty acid chains. And again, pure water has a density of 1. The density of seawater throughout large parts of the ocean is about 1.027. So triglyceride, 0.93 compared to 0.1027, is very buoyant. Provides a lot of lift. If you look at some lipids that you find in other fish, in the whales' heads, that different spermaceti organ in whales, and different types of lipids that are common in fish or marine mammals, they tend to store wax esters. And wax esters, they're really low density, 0.86. So that's the lipid that a lot of fish use. They store that in their bodies instead of triglyceride. It has the same result, is that for the same amount of volume, for the same amount of weight of some lipid, if you substitute wax ester for triglyceride, it doesn't seem like it'd make much of a difference. 0.93 reduced to 0.86, but it provides a huge amount of lift, hydrostatic lift, buoyancy, making that substitution. And then if you look at sharks, if you're getting rid of some of the triglyceride and substituting large amounts of triglyceride with diacylglycerol ether, which can be used for energy, just like triglyceride, and squalene, which is a lot more constant, not used, not broken down and cycled and used for energy so much. Look at the difference, 0.89 and 0.86 diacylglycerol ether and squalene versus 0.93. Again, it doesn't seem like it would make much of a difference, but it makes a huge difference in how buoyant this liver oil is, and the liver in general, and the shark, the whole body of the shark. So that's the typical kind of situation you find in deep sea sharks, that they have a lot of squalene and they have a lot of diacylglycerol ether. Here's a figure in the top, and inside that red rectangle, it shows these different species of deep water sharks. Now, if you look at a shallow water shark, all the way over there on the right in this figure is the sandbar shark. Typical shallow water shark, it's a typical animal, in fact. All that blue in that column above the sandbar shark is triglyceride. That's how most animals are. There's a lot of purple, light purple, that's free fatty acids. Now those are from the phospholipids chains, the three fatty acid chains that are associated with phospholipids, and some of them that have come off the triglyceride as well. But that's a typical shallow water shark. It's a typical animal, a lot of triglyceride. If you look inside that red rectangle now, you see a lot of yellow and you see a lot of magenta. That's what that color is. And in fact, if you add those two up for all those species in this rectangle, it's between 60, 70, 80, even 90 some percent of their lipid in their liver is either squalene in the yellow or diacylglycerol ether in the magenta. The blue, which is triglyceride, maybe it's 30%, but in most of these it's less than 20% of their oil is the typical animal lipid. It's not triglyceride, it's substituted by these other lipids, and those other lipids just happen to be very low density. Now, this squalene has been used, they make pills that you can take. Squalene is supposed to cure all kinds of different ailments and cure you from different diseases. So you can buy this squalene. These sharks are harvested in some parts of the world for their squalene. It's also used in cosmetics and high temperature and pressure lubricants. So if you look at the composition of liver oil in these sharks and you see, oh, well, it's got a lot of squalene and a lot of diacylglycerol ether in that oil. And the liver has a very large amount of oil. And the liver is huge. It's very big. The result of this is if you look at the density of the liver oil of deep sea sharks, the figure on the left here, these yellow species abbreviations, BSH, SND, CYP, those are all deep water sharks. And you can imagine a combination of squalene, which has got a density of 0.86, and diacylglycerol ether, which has got a density of 0.89, results in the oil itself having a density of about 0.88. That's really low density oil. And then it goes up as you get to these shallower water sharks, all the way up to sandbar shark, which has got a density of liver oil that's about 0.93, which is what you'd expect because it's almost all triglyceride. And the liver density itself, if the liver is big and it's filled with a lot of oil and this oil is low density, then the liver density itself is really low. If you look at the liver density on the x-axis, a neutrally buoyant liver is all the way over there at 0, where some of those squares are up at the top, some shallow water sharks. But all these deep sea sharks, those yellow dots here, have very low density livers. So it's negative in the parentheses. Those are negative, -5, -10, all the way down to -15. That is very buoyant liver. 15% more buoyant than water, less dense than the water that it's in. What that results in is that the shark itself, the whole body of the shark, is neutrally buoyant. So if you look at shark density in the y-axis of that figure in the black on the right, see these sharks are neutrally buoyant. They're not heavier than water, they're not lighter than water, they're the same density as water. Their density is 0. That's what results from having a big, oily liver with these low density lipids, is that the sharks themselves are neutrally buoyant. And that's what we see in species after species after species of these deep sea sharks. That is the prototypical deep sea shark strategy, although they're not sitting there planning this out. That's the characteristics you find in the majority of deep sea sharks. They're neutrally buoyant. Strong selection for neutral buoyancy. So, large liver, a lot of oil in that liver, high concentration of low density lipids. However, there's a lot of different species in the deep sea. And within a population of one species, there's males, there's females, there's young sharks, there's old sharks, there's medium-aged sharks, there's small sharks, big sharks. There's a lot of differences within species, between species, among species. Not everybody does it the same way. In the end, almost all of these sharks are neutrally buoyant, regardless of the species, regardless of whether the males or females, and regardless of how old they are, how big they are. However, as you might expect, there's a lot of variety within these different groups, and they all may be neutrally buoyant, but there's different mechanisms by which they obtain this neutral buoyancy. So that's what I want to look at in this second lecture, is how do these different species attain neutral buoyancy through different mechanisms? How do males differ from females? How do small sharks differ from large sharks? It's really interesting that in the end, they are all relatively close to neutral buoyancy. However, they go about it different ways. So that's what we'll look at in the second lecture for this Friday, April 17th.